Thanks for coming all the way to Davidson. I hope you had maybe an easier time finding it than we did. We didn't realize it was so close to the engineering buildings. It's my pleasure to introduce Dr. Fuqua today. He's a new professor in pharmacology and toxicology. He has, so he does research into the biologics and vaccine and microbicide prevention program. His background, he has a bachelor's degree in biology from Kentucky Wesleyan University. His PhD is from the University of Kentucky in Lexington, and he did his postdoctoral fellowship both at Wake Forest University in physiology and pharmacology, and also at the Owensboro Cancer Research Program. So he's been working on the project that he's been working on for a while, and now has become an assistant professor and joined us from Owensboro into the Louisville area. So yes, he's an assistant professor now, and also a big part of his responsibilities, in addition to starting up his lab, is program project manager for the U19. So there's a big grant in PharmTox, and he's helping out with that, being the project manager for this PREVENT program that looks at the biologic gerfitzin and how you can use that to inhibit HIV and HSV2 infections, and that's now going to clinical trials, and he's helping run that huge multi-million-dollar grant. In addition, he has a number of honors and awards and a lot of patents filed for his applications, and so you look forward to hearing his talk today. Thank you all for coming.
Thank you. So I was just going to introduce my lab. It started in January of this year, so I have one technician at this moment, and we have a lot of work to get done all the time. Right now we're exploring expression, manufacture, and application of proteins in plant systems. And plants in the wild, and then we can recombinantly express them, and then looking at how we can apply those. I also have certifications in regulatory affairs and project management, which allows me to work in interdisciplinary grants to help with those two areas. That's just outside of my actual research as well, more admin or regulatory affairs. I'm going to go ahead and give you the acknowledgements, because at the end, you get the conclusions, and then people want to kind of go back and give the acknowledgements, and I feel like it should be over when I give you my conclusions. And so I'm going to acknowledge these people up front, some collaborators here, Kenneth Palmer, Noe Matova, and Crystal Amorsky, and Lisa Rowan. They've all working with me and in our program, a PREVENT program, and developing an HIV microbicide. And then Karen McDonald and Daniel Toussey. I did a techno-economic analysis that I'll show you here, and they were both involved in that. And a lot of the work that I'll show you was funded by the Department of Defense or the U19 PREVENT program. And thank you all for showing up and listening today. So, what we're talking about is molecular farming, or basically manufacturing pharmaceuticals from plant systems. And really, it's just a means to an end, just like CHO cells or E. coli cells, but there's just some different subtleties that go about it with a plant system, and it's a much newer technology. There's several ways that you can get to manufacturing proteins in plants. You can transform them to express all the time. You can do transient expression, where they express it in a short term, but all of them end up in the same place. So if you're making a protein to purify to put into either, in our case, for pharmaceutical development. And this is just a list of, as of 2013, different plant-made pharmaceuticals and their development phases. And in this time, since then, this list has more than doubled. And with several of these moving on to phase 2 or phase 3 trials, and an additional product from this Protalix group has cleared the FDA. So there is some momentum in plant-made pharmaceuticals at the moment. So as I mentioned, there are a couple different expression systems, primarily looked at as transient or transgenic. Transient expression uses like bacteria or viruses to produce the protein of interest it hijacks the plant and uses them as the biofactory to produce your protein in a short term. Basically, you can go from gene to protein in about 2 weeks. You get high yields because it's driven through a virus or bacteria, but the issue is you have to handle your plants every time to infect them or inoculate them. Now, you can develop stable transgenic lines, which goes from gene to protein in probably more like 6 months, because you're actually having to integrate your gene either into the chloroplast or to the nuclear DNA. Then you can then it allows expression. It's generally constant protein expression. The great thing about them is there's less maintenance with that. You're not handling the proteins. You plant the plants, you grow the plants, they make the protein every time because it's expressing as part of their chloroplast or nuclear DNA. So there's advantages to both. Our group primarily uses transient protein expression. So that's the main differences, but I mean you can have stable and transient protein expression in CHO cells as well, which is a mammalian cell line used for antibody and protein production. Additionally, when we look at bioprocessing of plant tissue, it resembles very closely CHO cells as well. So right here I just wanted to show you a schematic of a few different lab scale and production scale bioprocessing models, in which you have CHO cells here in this beige color and plants in these green colors. You have bioreactors or fermenters that are producing your cells. These are then centrifuged. You have a couple filter stages, and then your purification occurs through packed bed chromatography. And then with plants, it's very similar, but instead of having a bioreactor, you're starting with plants on in a greenhouse. And so you have plants that are producing your protein, they're homogenized or ground, then you have some filtration steps, and then again, packed bed chromatography with filtration. And then at production scale, you're looking at homogenization through a screw press type device, a couple filtration steps, and continuous flow chromatography. So these are all, so the difference is, there's very few differences between the bioprocessing at the moment. Which brings me to one of our case study, or the molecule that we're going to be discussing today that I wanted to get into the bioprocessing of and things, which is Griffithson. So as Dr. Sabak mentioned, it's a molecule that we're developing as an HIV microbicide. It was originally isolated from marine lectin, and we produce it transiently through transient expression in Nicotiana benthamiana. That's like a tobacco plant, but it's a very old variant that doesn't get very big and is easily grown in greenhouses. It's not as big as what you may have seen in a field at some point in time. So they're closer to a house plant size. So we produced it transiently. We've tested it for activity against HIV. It has really great affinities to HIV and prevents transmission or neutralizes the virus. And thought is it would prevent HIV transmission in the presence. So when we discuss HIV microbicides, I just want to give you a little understanding. Sometimes that's a concept that people haven't heard of. So right now there's about 36 million people living with HIV globally, and there are 2 million newly acquired infections. There's no vaccine at this moment. You have a high cost of oral prep, even if you look at generics, unless they're heavily subsidized. And oral prep is an oral prophylaxis, pre-exposure prophylaxis. It's antiretrovirals that are given to high-risk populations. And so it's antiretrovirals that they would be on for as long as they're engaging in those high-risk activities, or as long as they want to be. So that's not without complications and costs for them. So the HIV microbicides are generally looked at as a transmission prevention mechanism. And they can be oral, they can be topical, there's even implantable devices that are being explored. Some of them are coitally dependent, they're daily dose, and some of them are even extended release. So there's no one size target as far as delivery and release. But the main goal is that they prevent HIV transmission, and they're likely to need to be very cost effective because of the markets that we're trying to enter with these would be, you want to reach as many people as possible, and they need to be able to use them without any hindrance based on cost. And the less you have to subsidize them, the more people you can reach. So we're going to circle back to Griffithson, which is what we're developing as a microbicide. And when I came into the lab, that we were looking at production of Griffithson, and the manufacturing of it at the time had used a pH 5.5 extraction with a ceramic membrane filtration, followed by a UFDF concentration step, SP chromatography, which is cation exchange resin, followed by RPC chromatography, which is a hydrophobic resin, and then a UFDF ultrafiltration, difiltration into PBS. So that was the final formulation. Problem with this is that it only had 30% recovery. The great thing about it, it was achieved a 99.8% purity. So this is Griffithson on an STS page gel. They're showing this is what the plant extract looked like, and then as it moved through each of these stages that I have numbered here. So you get a lot of it, it's really pure. It's great. 36% recovery is somewhat of a problem though, because if you're only recovering 30% of the total that was there, you're driving up your own cost of manufacturing. So the better you can recover that, you can actually help your costs. So what we decided to do was look at the physical properties of Griffithson, which are kind of unique. It's a 121 amino acid domain swap dimer, meaning that each one of the monomers takes a domain and swaps with the other monomer. So you can see that's why there's green in the blue here and blue in the green on this side. So this is one monomer and that's one monomer, but these are their domains that they've swapped with each other. There's no disulfide bonds, but there's a large number of hydrogen bonding within this molecule. So it has 3 high mannose binding sites, which are where its activity against HIV are found, so 3 per monomer, which is what helps it to be so active. The other thing is it has a melting temperature of around 78.8 degrees C, which is very high for a protein, and a PI of 5.39, but it is acid stable down to at least pH 3. And has very limited peptidase susceptibility. So even things like protease K, which cleaves a lot of proteins and things like that, is very difficult. Griffithson is stable against it unless you actually are denaturing the protein before you put it in contact with that protease. So what we decided, we would look at these properties and then try to adjust our bioprocessing off of that. Get ahead of myself sometimes. So We're going to improve recovery, reduce the complexity of the processing, but we have to maintain purity and activity. To do this, we're going to focus on those extreme stability characteristics that I told you about. So we looked at extracting from the plant tissue and then heating it. Very simple. We looked at 50 degrees, 60, 70, 80, and 90 degrees C. And we actually see the Griffithson stability of Griffithson is right here at 80 degrees C. And what has become our nemesis in this process is the coat protein of tobacco mosaic virus. Now, in the way we are inoculating these plants, or the way we get these plants to produce our protein is by using tobacco mosaic virus that is expressing Griffithson and then infecting the plants with that. It's a infectious virus that then replicates. And so this TMV coat protein is not in all your plants, but in all your plants that are infected and in producing your protein. So we have to deal with it or move to a different expression mechanism. So the take home here is Griffithson's still here all the way up to 80 degrees. We definitely don't want to go to 90. And the additional proteins seem to, well, the purity of the extract, even at 50 degrees, is immensely better than just at room temperature. So then we looked at this old paper, maybe, we started looking at, when we realized that the TMV coat protein